Good morning, ladies and gentlemen. It gives me immense pleasure in welcoming each one of you at this webinar on pharma possibilities of therapeutic monoclonal antibodies in partnership with Danaher Life Sciences and brought to you by Bluetech Media. At the outset, I would like to thank our partners for their invaluable support in helping us bring this much needed platform to you. In recent years, we have seen India make strides towards achieving better health outcomes, and this can be largely attributed to its robust life sciences industry. Historically, we have always been at the forefront of vaccine production and generic drugs. But the transition that we are seeing towards biopharma promises to be its most critical pivot yet, as India seeks to retain its position as the pharmacy of the world. As the industry matures and migrates from focusing on generics to one that puts R&D and innovation at the forefront, this promises to have profound implications for patients and industry peers. Now, more than ever, newer scientific innovations and advances are leading the way while bringing much hope for long unmet medical needs. One such vector has been the emergence of therapeutic antibodies, and more specifically, monoclonal antibodies as one of the fastest growing classes of pharmaceutical drugs. The incredible specificity, precision, and flexibility made possible by the use of MABS opens numerous avenues, and it is imperative that we understand its use cases and implications to the fullest in order to usher in an entirely new generation of therapeutic agents built on the premise of molecular targeting medicine. Over the course of this webinar, we will hear from industry leaders and experts who have their fingers on the pulse of this fascinating innovation and hear from them about the key trends shaping the space, as well as the innovation opportunities presented by it for industry practitioners. So, begin with, I would like to invite Mr. Anurag Tandon. He is the Vice President, METI and APAC region, Beckman Coulter Life Sciences, to address the audience and set the tone to this event. Anurag brings more than 30 years of multifunctional experience in his current role of Vice President, where he is responsible leading the commercial organization and driving growth. He leads the LS Council in India and has an extended role as a LSIG Director for India. He has worked with Beckman Coulter Life Sciences since 2016. Prior to joining Beckman Coulter, Anurag has worked in various organizations like Oxford Instruments, Mettler Toledo, Crompton Greaves, and led various functions like supply chain, product design, manufacturing, and P&L. So I'll hand it over to Anurag. Over to you, Anurag.
Thank you. Will be able to see my screen?
Not yet.
All right. So thank you, Ashish. And first of all, a very warm welcome. And thank you, Ashish and team, to organize this very important event today. And I think, as you've just described, monoclonal antibodies is gaining a lot of importance. And I'm really happy that we are able to be in touch with a topic of this sort today. And I'm sure a lot of people are going to gain. So I'm going to just introduce a little bit about Danaher. And as you may know, Danaher is a global organization. We are in the area of healthcare and life sciences. We are approximately about $30 billion of revenue in 2021. And life sciences does form a large part of this. So close to about $15 billion of our business comes from life sciences, which is kind of been for a few years now. So let me talk a little bit about the Danaher Life Sciences platform, which is what is our key topic today, and which is where we are very closely connected with the MABS development that's happening across the world. Now, here is a quick overview about what the Danaher Life Sciences deals with. So as you can see, we are approximately $15 billion of revenue when it comes to just the life sciences part of our business. Largely, the business is into biopharma. As you can see, almost 60% of our business comes from biopharma, which is, again, the topic today. If you look at some of our product mix, big part of the business does come from a recurring revenue, which is really a lot of consumables that many of our esteemed customers use. And about 30-40% of our business comes from capital goods, which are used as instruments and tools which are needed for development or manufacturing of biologics. We are very well spread out in the high growth market, North America and Western Europe, where high growth market is growing really fast. And there's a lot of investment that the organization is doing. Now, if you see the logos that you see on the top, these are well-known organizations in their own space, which are all part of the Danaher Corporation. And I'm going to talk a little bit about those in the coming slide. But each of them bring in a very unique element of products and solutions, which span all across especially around the biologics. Now, one of the reasons that there has been a gradual shift, and I would say what we call as a purpose-driven portfolio transition for Danaher over the last few years, and that's been more towards from life sciences, which used to be maybe about 30% of our total business, has moved to more than 50% now. So there's been obviously a gradual shift. And why is that happening? Clearly, we have some global growth drivers which are really helping the organization to move in that direction, both through acquisitions, but also through organic growth. Now, one part is really the shift towards biologics, right? I think there's a huge movement in the pharma industry where we are moving from small molecule to large molecule, and that's been a big driver in terms of investments and needs for the customers. Apart from biologics, we're seeing a large focus on the genomics medicine, and you will see some names which relate to that part of our product line of the applications. And there's future that is coming up a lot around the genomics medication. Vaccine and therapeutics research in response to COVID-19, I think we've been dealing with that challenge and opportunity in the last few years, the kind of demands, the kind of speed at which the solutions had to be given. And I think Danaher across various of these life sciences companies have played a major role in supporting many of our customers. And finally, we talked about high growth markets. I think there's a huge investment still going on into the basic and applied research capacity, which is where a lot of our business focus goes on to. All of the names that you see here, they are individual operating companies, and they have a leading market positions in their own space. If I move to this next slide, it basically talks about all of the names that you just saw, and I'm not going to go into individual details of each company. But on one side, you look at companies like operating companies like Beckman Coulter Life Sciences, which has a lot of precision instruments to study complex biological problems, and including causes of disease and potential new treatments. Then you have Leica, which is into leading edge microscopes, right? And that's again very well connected to the life sciences segment. We have SciEx, which is a global leader into mass spectrometry and again very widely used into the pharma industry. Then you have, when you go to the bioprocessing side, you have this unique combination of operating companies like Cytiva, which was XGE, and also Paul. And they bring in a very unique differential positioning in the area of bioprocessing, right? Where products from Paul and Cytiva combined, they give solutions across workflow of biologics and bioprocessing and various modalities. So there are solutions across from the cell culture and the single-use technology to chromatography to filtration, and also finally into the filling and fill finishing lines. On the other side, you have genomics that we talked about, a new area, and there have been two recent organizations which have been part of Danaher. One of them is IDT, which is really into developing and manufacturing nucleic acid, DNA and RNA projects. And the latest one, which is not on the screen right now, which is latest acquisition has been Aldebaran. And Aldebaran is a premium manufacturing partner providing critical nucleic acids and proteins which are used to make gene and cell therapies, and various gene editing technologies and DNA RNA vaccines. And it has this custom development and manufacturing services which gives the scientists around the world essential components to accelerate their research and develop reversionary treatments, which is what is the focus of genomics medicine. So, as we can see, over a period of time, there has been a purposeful shift, a transition towards more and more biologics. And I think we definitely feel that we contribute a lot in terms of the maps, which is what is going to be the focus area for today. And I'm sure there's going to be some really good presentations coming up as we in the next couple of hours, some from our own organizations and also from some of our well-known speakers. So with that, I would like to hand over back to Ashish for the next presenter.
Hey, thank you, Anurag, for the insights. So now before we proceed further, I would like to inform all our participants that we will open the floor for question and answers at the end of the session. You just need to click the Q&A button at the center of the screen below, write your question by clicking Ask Questions, and we'll take this up with respective speakers. Moving ahead, we would like to welcome our next speaker, Dr. Narendra Chirmule, co-founder and CEO, Symphony Tech Biologics. And he will be talking on exploring the future of monoclonal therapy. Narendra Chirmule is the co-founder and CEO of Symphony Tech Biologics, a data analytics company focused on engineering solutions to biology. He is the chief scientific officer of an immunology research services company, Acuimmune. As former head of R&D at Biocon and leadership position in Amgen and Merck Vaccine, he has contributed to clinical development of vaccines and biopharmaceuticals. He did a PhD on development of a leprosy vaccine from Cancer Research Institute, Mumbai, postdoctoral studies on pathogenesis of AIDS from Cornell University Medical College, North Shore Hospital, New York, teaching and research as assistant professor in gene therapy at University of Pennsylvania, Philadelphia. Dr. Chirmule is on the NIH Advisory Committee for HIV vaccines. So over to you, Dr. Chirmule.
Thank you, Ashish, and thank you, PU, and everyone else who helped organize this conference or this webinar. I'm delighted to be able to talk about monoclonal antibodies as therapies, and let me just open my slides. I believe you can see my slides now. Okay, so I think this webinar is going to be on antibodies as drugs, and I assume the audience here is probably familiar with antibodies, so I'm not going to go into the basics of antibodies. Sufficing to say that they are very complex, large molecules, 150 kilodalton, and many aspects of the antibody are described here. The light chain, the heavy chain, the CDRs, complementarity, determining regions, which is the regions of the antibody that bind to antigen. Antibodies are quite well characterized over the last several decades. The FcRn binding site, which is shown here in orangish pink, is one of the key aspects of the antibody molecules which distinguishes it to make it a fantastic drug for therapies. And the reason is because Fc neonatal receptor recycles IgG. So when Fc portion of that FcRn portion of the antibody binds to the Fc receptor, it is taken up by cells like endothelial cells, etc. It goes through these vesicles, and through a pH-dependent mechanism, the antibody, instead of being degraded like many other proteins that are degraded in the cells, antibody molecules are recycled back into circulation. This property of antibody is the one unique feature of this antibody molecule therapy that makes it a unique therapeutic agent. And try to get to the next slide. Yeah. And this gives it a very long half-life, sometimes in days, sometimes in weeks, and sometimes even in months. Denosumab, for example, is an antibody that is given only every 6 months. They're even trying to give it every year. So you can see that these antibodies are very, very good targets, and make a very good therapy because they have to be given so infrequently. So the success of antibody therapies has been multifactorial. As I mentioned, the FcRn interaction gives it a very long half-life, and as Ashish and others have mentioned, that it is very specific. It has limited off targets. A lot of formulation studies have been done to make sure that it's more stable. And we understand quite a lot about the manufacturing properties of monoclonal antibodies now. So it has a high risk benefit ratio. The ADME properties, which is administration, distribution, metabolism, and elimination, are very favorable for making it a fantastic drug. Often non-linear PK, various modes of administration, low dosing frequency, as I've told you before. One sort of challenging feature of antibody molecules is because they're so large, they have poor distribution into tissues, especially intracellular targets. They are metabolized usually by unspecific proteolysis, and they are eliminated through molecular weight dependent renal filtration and receptor mediated endocytosis. And their pharmacokinetic profile is often dependent on the PD effect, pharmacodynamic effect of the antibody. Next slide. Yeah. Over the last 3 decades, making monoclonal antibodies as therapies has advanced a lot, from fully murine antibodies to now fully human antibodies. And there are now many technologies, many different kinds of technologies that enable this fully human antibody and reduce the level of immunogenicity. That is one of the major side effects of monoclonal antibodies. Characterization of the molecules through understanding product quality, critical quality attributes. Monoclonal antibodies are very large molecules and therefore have many, many components to be characterized, like the deamidation sites, oxidation sites, glycosylation sites, truncation sites. So you will hear a little bit more about all the analytical instrumentation and the techniques that are required to measure all of these parameters. Like I said earlier, immunogenicity is an issue that monoclonal antibodies have to overcome. And as you can see in this slide, which shows a time kinetic of time versus concentration of drug, when you give monoclonal antibodies and there is no immunogenicity, you see the black lines. They last for a very long time. However, when people or subjects develop antibodies, or neutralizing antibodies, or clearing antibodies to the drug gets cleared a little bit faster. So this is a very important and important feature that needs to be addressed in the development of antibodies. The pharmacokinetic profile, there are multiple ways that the drug is cleared through central clearance, drug-mediated clearance, and also through anti-drug antibody mediated, immune complexes mediated clearance. So very clear understanding of how these drugs perform in patients. From a toxicology perspective, the toxicity that is seen in monoclonal antibodies is very different from small molecules. From small molecules, generally, predominantly you see liver toxicity, maybe lung toxicity. But monoclonal antibodies have a different array of general toxicities, sometimes on target, some very rarely off target. But sometimes you see things like platelet activation, pancreatic fibrosis. Agonism is a very important aspect of antibodies. Platelet activation and vasculitis. Generally, they have a very big um, treatment window. Again, a lot. This slide contains a huge amount of information, just shown you in a very big summary. Over the last 30 years or so, large molecules, which is monoclonal antibodies predominantly, have been terminated. Development has been terminated for various reasons. And I just give you some percentages of some data that we've done a systematic analysis on. About 28% have been killed in toxicity, 44% due to lack of efficacy, 16% due to pharmacokinetic and immunogenicity issues, 6% due to adverse events. So you can see the number is very small for adverse events, and 16% due to manufacturing challenges. This is just to note that 50% of the molecules may be terminated not for biological activities of the molecule, but more for business reasons. So this gives you an idea of the kind of reasons monoclonal antibodies fail drug development. The main point to note is that very few due to adverse events. Next slide. Yeah. So if there are what are the limitations of monoclonal antibodies as therapies? Right? So some of them are listed here, and I'll give you some examples of how they can be overcome. So the first one is efficiency in killing the target. So native antibodies depend on antibody-mediated or complement-mediated killing to induce killing mechanisms. So that the ability of antibodies to kill molecules, to kill their targets, can be enhanced by conjugating them with toxins. Monoclonal antibodies have single specificities, and now multi-domain antibodies and multi-domain biologics are being made to overcome that single specificity. Agonism, which is when an antibody binds to a target, induces a positive signal, which sometimes is detrimental. It could be an off-target side effect that you could observe, like T-Genero is a good example. To overcome that, single-chain antibodies and hemibodies are being designed. Antibodies already have a very low clearance rate, and that clearance rate can be further decreased by making catabolic antibodies and modifying FcRn-related functions. Specificity is not an issue generally, but you can increase specificity even further by making mimetics and avimers. One challenge with the antibodies, major challenge, is to have intracellular targets. And so small molecules or nucleic acid siRNA can be delivered through antibody-mediated targets, conjugated targets. So these are some examples of how beyond monoclonal antibodies, what can be done using this technology. This is just an example of the different kind of antibody fragments that are being designed to make therapeutic proteins. As you can see, most of them, if not all of them, retain their FcRn function, which is the predominant factor for antibodies. And then more recently, almost 10 years ago now, the use of antibodies to make CAR T cells using the SCFV fragments and the CD3 cytoplasmic tail are also being generated. This is a nice paper, which I've put in the link here, which shows a lot of different kinds of antibody and antibody structures that are being made for making multi-domain, multi-specific antibody structures, which is going to be the novel drug development of the future. So in general, I'm going to summarize finally by first pointing out the 5 points that I made earlier, why monoclonal antibodies were so successful. There are 5 more reasons with all the new technologies that are being developed for making it even more successful, by having even more infrequent dosing, by increasing the specificity, by making them multifunctional, by making them more safe, by having many features. Formulation will make them more stable, and manufacturing from batch mode has gone to continuous manufacturing, and therefore much advances are being made in the in those technologies. So I'll stop here and I'll hand it over back to Ashish and the team to see if there are any questions, and we can take it from there. Thank you, Ashish.
Thank you, Dr. Chirole. As I mentioned, we'll be taking the questions at the end of the session when we'll have all our speakers in the studio and we can take the questions one by one. Yeah.
Excellent.
So
Thank you so much.
thank you very much for your wonderful presentation. So moving ahead, let me welcome our next speaker, Dr. Neeti Jetli, application scientist, molecular devices, and she'll be speaking on future of therapeutic monoclonal antibodies. Dr. Neeti Jetli is currently working as a field application specialist for molecular devices. Before this, she was working for a decade as a group leader at TCG Kolkata, a CRO organization working for drug discovery projects in inflammation, pain, and oncology predominantly. Being a former customer helps understand customer needs and making sure their applications are met with. She has done a PhD from ICMR in biochemistry, followed by a postdoctoral fellowship at Northwestern University for 3 years in molecular and cellular biology. So over to you, Niti.
Thanks, Ashish. I hope everybody can see my screen. Okay, so I will be presenting on future of therapeutic monoclonal antibodies. And and first of all, I would like to thank, before I move ahead, a wonderful presentation by Dr. Naren, and that really formed a beautiful base for my presentation, and I can connect very well now. So first at Danaher, Anurag gave a fantastic introduction to what is Danaher all about. And at Danaher, we constantly work with continuous improvement to help our customers. And this workflow, which I'm just showing you, monoclonal antibody, internally with the team effort, talking to our customers, we made these workflows so that, this is for monoclonal antibody, we have similar workflows too for other techniques. But if you see this, there are these 6 processes, like broadly classified: monoclonal antibody designing, characterization, transfection, cloning, production, harvesting, purification, right till FDA quality control. And down below, you see all our opcos, operating companies as we call it, under the Danaher hat. So under one hat, you're going to get all these offerings. And then also we kind of have a glimpse of where we requested all our opcos. We work with a great team effort, and we align the products which go into each of these techniques. And this really helps the customer understand that, okay, what am I looking for? What kind of antibody process I am in? And then align the products accordingly. And that's why we made this internally also. So it is good for our understanding and to help our customers more efficiently. And then further, we deep dive into these monoclonal antibody workflow. So you take that one process, like antibody designing, what would it have? Screening for binding to specific immunogen, immunogen characterization, assay development, cell-based imaging, what kind of techniques they want to see for the functionality of the antigen-antibody reaction by which they would, design the chimera or whatever kind of monoclonal antibody approach they want to go in. And we can align the products according to which stage they are in the monoclonal antibody production. So after giving you this brief monoclonal antibody workflow, we would say, okay, why are we concentrating more on, monoclonal antibody? The question is, as Dr. Naren beautifully explained, that why monoclonal antibody, is a very, it forms like a good drug target. And if you see the global market, because, we always keep an eye on the global market, that how is it going to move from now to like 10 years or 8 years from now. So if you see North America, Europe, Asia, or even Middle East and Africa, you can see the trend of the monoclonal antibody increasing. Another research and markets, when I saw, I was really happy to see this, that from 2021 to like 2028, we do see a market increase in the monoclonal antibodies. Further, if you see that, okay, what part, like oncology, immune-mediated disorders, infectious diseases, cardiovascular, neurological disorders. And well, this was taken from a paper of like 2020, so we do not have one more in incorporation of corona and COVID-19, where it has changed all our lives, and even the way the research is moving ahead now. Because so this gives you a snapshot on how monoclonal antibodies. So if you see, there are 131 monoclonal antibodies being studied in all these areas. Then further on going, I was getting, when I was reading, I was getting a lot of papers on what is the global drug sales in the top 100 in 2020. And I got this list. And of course, the list was very long, so I just took a snapshot. And we can see that how the importance of monoclonal antibodies are increasing. And for that, this workflow, which I just presented in these previous slides, help you to align in which process our customers are, and to help innovate technologies. If you see from 10 years back from now or 5 years back from now, the way we used to do research definitely changes. And that changes because of you, our customers, and us working in together to get more innovative technologies for a faster and a better result and research. This is just a schematic representation I really liked of AstraZeneca. Process of discovering and developing an antibody-based treatment, because we know that they are making the COVID-19 MAB. And this is the source for the antibodies against the SARS-CoV-2 virus. So, if you see on the left, patients who have recovered, immunized mice immunized with the spike protein, laboratory techniques such as fast display, as we know, that is also one of the techniques through which monoclonal antibodies are, characterized. Harnessing multiple technology platforms. The second one, immune replica technology, hybridoma technology. There you see the B cell sorter, and then, you can see the monoclonal antibody getting generated. Screening to assess the characteristics of the potential monoclonal antibodies. So how are you going to do that? The binding, of course, you want to see what's the KD, what's the dissociation constant, or ELISA, how specific it is, and neutralization, that how much is it going to be used to neutralize and to get the required therapeutic effect. Developability, that is very important too. And then testing, development, approval, and manufacturing and distribution. So, this is the whole workflow where, we work along with you guys so that, we can align the products used and work towards innovation. So this is a snapshot going further on of mouse hybridoma, phage display, transgenic mouse. So whenever we approach our respective customers, they have different approaches, of course, because everybody is working on different therapeutic areas and have different approaches. So this is in a nutshell just how the antibodies, therapeutic monoclonal antibodies are processed at the inception till getting it to the characterization stage or the functional screening stage. And so if you see here, the phage display or the mouse hybridoma, so harvest splenocytes, generate hybridomas. Then once you talk about screening, where you have your clonals, you have thousands of colonies. How are you going to screen that? We all know we used to do, single cell limiting dilutions, but so when you have automation there, you have instruments there which can speed up. Then there is phage display, where you can even improve the screening time. And then, of course, the purification techniques, where you want the best purification technique, where you get the impurities getting rid of, and so on and so forth. So going further on with this snapshot in mind, the technologies used predominantly for monoclonal antibodies. So we want clones with monoclonality assurance. So on the right, you see that we have, instruments for those two, where it can give you monoclonal assurance, that is monoclonality assurance, right, when you're cloning, when you're screening for the clones. Then we have cell sorter technologies where B cell you're going to do the cell sorting and then figure out on getting the monoclonal antibodies. Then we have these purification methodologies. So when we say we have the leading brands like Paul Phenomics, or we have our other leading brands, and we can see here how they can help get or the required purified product like LCMS, MS, as Dr. Nareen mentioned about PKPD. So you want to study the metabolites, and here you can, easily get those. Here again, when you want to do functional screening, we have products, microscopes, like manual, semi-manual, or even high content systems where you can easily, because we all are approaching towards 3D, and when you want to see 3D T cells, CAR T therapy, that is also another approach which is going to come in. And this is I like taken from a reference, and customers are using this and finding it, a great asset. These are just a snapshot of all the technologies. Again, just techniques being used. Why I'm, harping on this is again that how it is going to accelerate the whole workflow, which I just showed in the previous slide. And you can see, apart from all these techniques like cell line development, binding affinity, viral neutralization, when you talk about, when you go talking about COVID-19 process flow, viral pathogenesis and viral titer, cell count, cell viability, and then purification techniques, of course, cell sorting techniques, ADME, PK. So under one hat, we can help you in all this. All automate your workflow. So this is one very crucial thing also which customers look for after the pandemic. We have seen customers look more for automation, that not a single plate, but we want to do like say 20 plates or 30 plates, and we can help you automate that with our esteemed team and work along with you. So this is the whole workflow of the monoclonal antibody production, right since the inception of creating a hybridoma till expansion stage. And whether you want to automate partially or the full end-to-end solutions, our whole team can help you do that. And this is my final slide where we talk about what do we take away from this whole thing, what we were discussing till now, is the field of therapeutic antibodies has definitely undergone a rapid growth in the recent years. And we are seeing that through, global market and the kind of monoclonal antibodies being used already in the market. Then we have antibody drug conjugates and cancer treatments from, predominantly we know that for cancer or autoimmune disorders, it's broadly used. And targeting the tumor microenvironment or targeting immune checkpoints. Then coming to, in the future, studies evaluating synergistic effects of antibodies and chemotherapeutic drugs, or biomarkers and antibody-based therapy for human diseases. So that is very crucial, and we can do it with this monoclonal antibody approach. Second category of antibody drugs would be to enhance the therapeutic value. We have heard about the CAR T therapy, where your T cells, along with the cancer cells or the chimeric antigenic receptor with your cancer cells, they are being used, and how bispecific antibodies or antibody radionuclide conjugates would specifically get that molecule to the specific area of interest. That would be really the in thing in the future too, seeing to what the recent papers and research are talking about. So this is all about the future of therapeutic antibodies, and thank you for patient listening.
Thank you, Dr. Niti, for a wonderful presentation. And while we wait for Dr. Ratnesh Jain to join, we'll take a couple of questions that we have received from the audience right now.
Sure.
Okay, so the first question is, can you share some light on how are you going to handle the complete solution offering from Danaher, uh, being the Danaher bringing solutions?
Yes, so that's a very good question. And so we have, formed groups within Danher, and we are trying to uh, go towards how we can meet the customer needs. And like we have initiatives, and we are working on that. And it, yeah, for a smoother transition.
Okay. Uh, there's one more question that we have. Could we automate clonal screening with monoclonality assurance?
Yes, absolutely. Because when I interact with my customers, and they talk about that monoclonality assurance is one very key thing, which we look when we are screening. That's why the name is monoclonal antibodies, and that's so you can absolutely do that. We have instruments for that.
Okay, thank you very much. So by the time Dr. Jain is joining us, he's a bit late for a couple of minutes. I'll get Dr. Chimule as well on the screen, but then we have a couple of questions for Dr. Chimule as well. Hello, Dr.
Okay.
Chimule, once again.
Yeah, hi.
Okay. We have one question for you. What are the current major trends that you're witnessing in the various segment of the industry? Also, if you could tell us about the possible future trends.
Yeah, I think it's a difficult question to answer what the future is going to be. But the future will, as I mentioned, all the factors that I mentioned, antibodies have deficiencies for, like the increased specificity, increased fewer injections, so that you can get longer duration of the treatment, multi-specificity, which is a very big factor which is coming up in the future. No disease can be treated with one drug. You need multiple drugs. And therefore, having molecules which can target multiple pathways is going to become the future. And I think very interesting molecules are coming in the pipeline. 2, 3, 4, 5 specific antibodies, not just antibodies conjugated to many different things. In my mind, I call them like monster molecules. They will have their own set of challenges, but that's where the field, I think, is going.
Okay. We have one more question. How do you balance the need to respond to a crisis like COVID-19 rapidly, while at the same time keeping a long-term resilient course?
I think COVID has taught us a lot of things. One is collaboration. I think unprecedented level of collaboration is what made COVID vaccines happen. I think the readiness for manufacturing at scale, all of those were very, very important aspects of the process. I've written a book on this, and a chapter on COVID-19 vaccines, and why the world could develop a vaccine in less than 1 year when normally it takes 10 years. And there are many factors regarding that. I think one of the important points is to be to sort of miniaturize the process, make sure that you can actually make high yield product, make cell lines which are making very high level of protein potentially, 10, 20 grams per liter. I know it's difficult to imagine that today, but things like that. Yeah. So Niti, maybe I'll ask you a question.
Sure.
Yeah, how I mean, I know you talked about a lot about instrumentation and how instrumentation can come together and make efficient. Do you think that we will ever be in this situation where pharmacies can ever make their own drug on demand?
We are working towards that, trust me, because earlier, Dr. Naren, as like 10 years back, when probably I would have done my PhD, we were doing everything manually. Now when I look at this automation, like we have an automation where end-to-end solutions, where you just put a plate in, and at the end you get all the data with you and your hand. So where that is concerned, yes, so that I don't have to wait for a month again or 15 days later to get my clone, or to purify that and then to go. So I am seeing that 2 months being decreased to like, a few days or like, so I'm hoping that, you can do it on demand.
Yeah. And the other thing related to antibodies is to generate the antibody. Right? You have an antigen that you want to develop the antibody to, and then you have to
Absolutely.
generate the antibody. That usually takes 9 months because you have to immunize mice, make monoclonal antibodies, make hybridomas.
Yeah.
That technology has to be disrupted. And any
I
development
agree.
in that area that you are aware of?
Not that I am aware of, because still they do immunize in the mouse and do get the, finally the fusion part. So we need to really work on that part also, where you can get. So I guess this part, even like, you synthetic biology, we would have heard the name of, but then it's too broader term, and we need to see results in that synthetic biology. When we say I can make a molecule, but then like, I was in peptide synthesis, so I made peptides, but then are they going to be identical to give me a same KD value, or the same IC50, or the same kind of ADME properties? So that, yes, it seems very nice and good, but we need to see the biology too when it comes to that.
Yeah. Prachi, I like your question actually. Is are Indian companies ready to enter the novel therapeutic discovery and development market? I think absolutely, absolutely. I really very feel strongly about this because
Absolutely.
I think there are many factors that have brought together where we are in India today. I think maybe 25 years ago, when Indian companies started developing generic molecules, companies like Cipla at that time, Ranbaxy and others, uh, it was a very interesting phase where the scale of the small molecule development became very, very high. And now we are the world's largest pharmacy for small molecules. Then about 10 years ago, I would say, the whole process of biosimilars started being generated. And so when biosimilars were generated, today, there are so many companies in India who are developing large-scale biosimilars, and we are in the process of that growth. And I think the next obvious phase is going to be novel molecules. So the way I look at it
Yes.
is when we develop generics, we were in elementary school. When we are developing biologics, biosimilars, we are in high school. And now अब कॉलेज जा रहे हैं, सीखेंगे सीखेंगे, पीएचडी तो बहुत देर में होगा. अभी तो कॉलेज जाएं. So that is the
Absolutely.
progress of how we are in novel drugs. But we will get there. But we will get there faster than we can imagine. And I think in the next 5 years, there'll be an explosion of novel molecules in India.
Thank you
Absolutely.
very much, both the speakers. Now we have Dr. Ratnesh Jain with us, and we'll continue with the next session. Thank you. Please stay, and we'll continue this Q&A round after his session as well.
Yeah. Thanks, Ashish, and thanks, Dr. Naveen.
Okay, so let me welcome Dr. Ratnesh Jain, Head and Course Coordinator, Bioprocess Technology, ICT Mumbai, to share his views on the topic Analytical Characterization for MAB Development. Dr. Ratnesh Jain is a recipient of BIRAC Innovator Award, Humboldt Fellowship, and Ramalinga Fellowship. Dr. Jain has demonstrated industry-academia collaboration and have completed more than 50 sponsored industry projects in last 10 years. He is also founder of VTranslate and co-founder of Away Bioscience, a 3D printing startup jointly founded by IITM and ICT alumni. Dr. Jain is also leading skill development and training program and organizes one of the largest biopharmaceutical hands-on training program called Biosimilar Workshop. So over to you, Dr. Jain.
Thank you, Ashish. Thank you very much for the kind introduction, and thank you, everyone. I hope Naren and Nidhi has given quite a good introduction to what antibodies are and how these antibodies, specifically the therapeutic antibodies, are needed. So, just listening to what Naren says, that it is going to be a very promising future, which we are looking at it. So, allow me a moment where I'll demonstrate my slide. Um, So Ashish, if you could confirm my slides are visible to the audience.
Yes.
So today we will be talking about analytical characterization. So as you have got a good glimpse of what antibodies are, how these are typically developed, and I'm going to largely focus on how these antibodies are characterized, which is a very critical part of the entire development strategy. Before I begin, quick introduction of who am I and what about the Institute of Chemical Technology. It's one of the oldest institute, elite institute of western part of country, largely focused on pharmaceutical and biotech programs, which are very prestigious program of our country. We are also having a lot of motivation from our alumni who are, motivating us, inspiring us to doing something good for the society and encourage more industrial translation of the product which we develop in academia. About our nanomedicine research group, which is largely divided into two parts: farm biotech, where we look into upstream downstream part, and biological characterization, which is headed by me. We look into analytical and bioanalytical parts. um, Coming back to biopharmaceuticals, so what are biopharmaceuticals largely, and where we will find antibodies? So it's a large set of molecules from oligos, peptides, cytokine, also different kind of, just a minute, I'll add laser pointer. So if you could look into, so cytokines, recombinant proteins, which is largest category of drugs you will see. Another set of categories which are coming is fusion protein. Most of the upcoming research is coming on bispecific antibody, antibody drug conjugate. You will also see a lot of vaccination strategy like DNA vaccine or whole viral vector based. MAPS is the current focus for today, which is one of the leading therapeutic strategy. Again, for a niche area, for niche therapeutic segment where antibodies are developed, and gene therapy, which is also developed significantly, but Indian perspective, it will take quite a good amount of time before it reaches to the common patients. But yeah, of course, considering this, peptide and antibody for Indian population is one of the largest segment where development can be rampant. And that is something which is also one of the focus area where large major companies are working in. If you look into certain old data set, a lot of antibodies are value creator, which you could see. So these are some of the value creator in 2024 which has been predicted. A large amount of value creators are antibodies. Now you will also see these are certain 10 areas predicted back in 2018. Anti-rheumatics, anti-diabetic, which is also one of the major area, antivirals. And I would say this number will definitely, because of COVID, now antivirals are the primary important. It could be much bigger market shares they can occupy because of COVID. These are again certain we very well know in various presentations you might have heard adalimumab, which is one of the largest selling, but there are many others. So pembrolizumab, we don't have even one pembrolizumab in Indian market so far. But that is going down around 2026, 2027 when largely a biosimilar can come for this particular. We have many because of COVID. You also see this particular duplimab, which is uh, to understand certain concepts, specifically COVID-related symptoms. Duplimab is ustekinumab, which is also anti-IL-12, largely in the development phase, which you find. So these are, and specifically, I would like to bring your attention to the large 14th one, denosumab. So denosumab and teriparatide, which is another peptide, is largest focus. So COVID has given a lot of osteo symptoms. And these symptoms, which are largely relevant to osteoarthritis or arthritic symptoms, or specifically bone weakness, and largely, though there is no proof yet to be given by clinical trials, but denosumab and teriparatide, these are doing extremely large amount of business or large development is needed. And this number will actually go up in the current scenario as well. Now, if one has to look into biologics or biosimilar growth strategy, these are biosimilar approval will continue to increase because now we have much greater clarity from FDA review process. Most of the companies are entering into this domain. Biosimilar litigation is also booming. It means the companies who are having patent security, they are launching a lot of litigation, which will definitely allow more biosimilars to come into the market, again for the patient compliance as well as the affordability to the patient. Again, education, which we are largely part of that, it will also speed the market uptake. So education is, and India is actually leading in terms of biosimilar education and most of the biosimilar awareness program, which is happening all around the country. Certain guidance, why do we need and how do we come up into that? So there are certain guidelines which are ICH and Q. We largely handle the structural basis of the antibody. So what we are looking at here is how do we understand primary, secondary, or higher order structure. Most of the time investment also go to look into PTMs, post-translational forms, biological activity, purity, and also immunochemical properties. All the ICH guideline Q6B for stability and other things, we are looking into structure. So when you look into biosimilarity, structure plays in very critical roles. Look into where biosimilars are going to differ. If I'm going to have a biologics, a new drug, which is coming as a biologics development or a new antibody, I'll be spending a little time in analytical, but I'll be spending most of the time in the clinical safety and efficacy, because here a new drug and its receptor plays an important role. Its biological small molecule are even going for bio-waiver. The reason is because biological or clinical trials are not needed, analytical similarity is enough. Considering that protein or protein molecules are not same but similar, clinical things is still yet to be reduced or elevated from the approval process. So analytical, and here we play a major role to understand how analytically we understand these antibodies. So drug development can be faster, antibody development can be faster. This is a motivation. We are looking into how do we understand the structure of protein molecule. Again, some key differences. What we are looking into a biosimilar and generic. So when you are looking chemical small molecule, and complexity is huge. Previous speaker might have given this glimpse, but the development cost of a biosimilar is almost 100 to 200 million dollar, which is huge cost compared to a $1 million generic development. You also need to spend 8 to 10 years. Though COVID has given a lot of boost in the entire development process, but still, timelines cannot be defined. And when you are looking for more establishment of a therapy, time is one thing which one has to give. And when you are looking into the chemical small, so these are very less complex, robust processes are there. But biologics is like Titanic. So this comparison is very simple comparison of a Titanic to a bicycle. A bicycle you can understand much better, but a Titanic, if you need to understand, there are a lot of complexity. So we have a complex production product antibody. This is a fragile product. Product can be degraded. Process is highly variable. Process losses are very high, and there is very, very high risk of contamination. These problems basically comes to the development process. That is the reason you spend more money here. These are the also that you spend more time here to understand these molecules, and the risk associated with adverse effect or the risk associated with adverse understanding can be also very high. So where do we need analysis? This is a simple antibody production process. If you have a cell culture, you put it into certain reactors, do your filtration upstream, downstream, bioprocessing, viral filtration, you get final biopharmaceutical, final antibody here when you start. So this entire process, but analytics comes everywhere. So you have upstream process, you require analytical understanding by whether how much is aggregate, what is the glycan profile, what is the purity of antibody. In downstream, again, the same thing, aggregate, host cell proteins, protein achromatography, purity and yield. Also, in the formulation development, aggregation, secondary structure, higher order structure, because when you put into the formulation component, there is a great impact on secondary and higher order structure and purity. So analytics comes from the very beginning till end, and analytics define the quality of the product. And when I'm developing an antibody, what should I look into? So there are some global understanding. Global means a larger. So what is very critical is mass. What is very critical? So you need a mass spec. You need to understand shape of the antibody. You need a light scattering, SBAUC. You also need a solution state understanding, charge of the antibody. Then it goes to tertiary and secondary structure, which basically need to understand conformational stability, colloidal stability, tertiary and secondary structure, which is done by the methods indicated here. At the residue level, you need to understand glycosylation, PTMs, degradation, and these are very important. So this is the typical attribute you need to go understanding globally till residue level. And also there are many other things which are like functionality, size, charge, identity, which by multiple other methods. All the excipient has to be characterized, and also packaging like leachables and extractables. So antibody product development is not a standalone development. You need to understand holistically. A 360-degree approach is needed to understand analytical processes. If one has to develop the entire analytical understanding process to demonstrate a product which is qualified as a therapy. So, so this is, some quality attribute. Now, if you look into a protein, you have multiple, as I'm consistently saying, there are multiple issues. It's very complex. So what you are looking into, these disulfide shuffling. You are looking into aggregation, because protein, naturally, as per the fundamental rule of thermodynamics, it goes to the minimum energy stage. And the minimum energy state comes only, when they are most stable, and that is the reason they aggregate. So aggregation will happen. You can't really restrict the aggregation. You can only control the aggregation. So one has to understand the controlling strategy as well as analytical process to understand level of glycosylation, level of fragmentation, truncation, deamidation, different pyroglutamic molecules. These are almost more than 100 attributes are characterized over the period of time. And these are certain methodologies which are used for these characterization processes. LCMS is also one of the leading methods to understand all these, specifically in tech mass, fragment, PTM, and glycan, to understand. But it's not only one method which is going to justify, but you need multiple methodologies, which we call orthogonal approach, to justify each and every process. Analytically, we need multiple requirement. We have structural requirement, primary higher order structure or molecular weight. You have functional requirement like bioassay, target binding, FC receptor functions. Product, when the product is there, again, aggregation, oxidation, charge, glycosylation needs to be characterized. The method has to be state of the art. It has to be orthogonal. It is convincing enough that whatever claims you are making that my antibody structure aggregation is so and so, these methods must be complementing each other, because one method will not be able to capture all the data you need. Stability is pretty critical here. You need a degradation profile of different stress. Specifically, two things comes very critical here: temperature. All these molecules are temperature sensitive. Their storage is at 2 to 8 degrees Celsius or sometimes less. It means they are going to experience freeze thaw higher and lower temperature, and that also impact the quality of material. You also have impact from light and agitation, which sometimes agitation aggregate material, and pH, because most of the antibodies are stable in pH buffers. And if pH changes, their secondary structure or sometimes even the nucleation, which is initiation of aggregation, can begin. So this plays an important role, and one has to demonstrate the stability there. Impurities, impurities, specifically aggregate impurities, the injectable products. So one has to look into subvisible particle, and also process. It is going into multiple downstreaming process, starting from protein A chromatography to multiple other chromatography stage viral, it means the process impurities are going to bound to become. Since the host is always a host cell or a CHO cell or any other type of cell, host cell protein comes and they can be very allergic. Media components, which are largely utilized to grow the cells, to get the product out, can be one of the impurities. So these also needs to be understood. So as I'm telling you that as a product, you need to have a comprehensive requirement analytically, and you should have a comprehensive strategy to demonstrate that antibody development will only be completed once your analytical methodologies are established and they are demonstrating safety as well as the structural integrity of the product. So, from the reference product to biosimilar candidate, largely what you need to do: characterize reference product. You need to understand CQAs of the reference product, critical quality attributes. You should have your unique cell line, which is also one of the major chunk of. If you do this particular step right, the rest of the step will actually eventually be right. But this is one of the most critical steps. So if somebody is developing a biosimilar antibody for the reference antibody, this step has to be extremely critical. And once you pass on this particular characterization and identify whether your quality attribute matches to the reference quality attribute, and then evaluate similarity. And so here the question will come largely: what are the best analytical strategies to develop antibody product? So with this question, which will remain in your mind, that if I'm developing a biosimilar, what is my analytical strategy? I will end my presentation and we can discuss more if you have more questions about analytical strategy to develop antibodies. Thank you very much. If you would like to learn more, we arrange digitally biosimilar workshops, free to attend digitally. So you can feel free to visit our YouTube channel and learn more about, antibody development. Thank you very much. Thank you for the attention. Thank you, BlueTech and Ashish. Thank you.
Thank you, Ratnesh. Thank you for this wonderful presentation. And I would like you to please stay there. We'll add our other speakers as well, and we'll open the session for Q&As. Hello, everyone. Once again, welcome. And we will continue with the questions that we've already received from the attendees. This is for Dr. Naren. While most of the MAB discovery has been based on the Caucasian genome library, Indian genome presents a much wider CDR3 region. What can be done to explore the Indian genome much better? Also, we really don't see any novel discovery happening in India. This is from Mr. Vivek Chandra.
Yeah, I think I already answered the question about novel discovery in India. I think as far as the genome sequence, CDR genome sequence in humans is concerned, it is pretty conserved, even though majority of the antibodies libraries that are right now generated may be of Caucasian descent. I think there is still a diversity that can be generated from other libraries. I know, for example, in India, Zumator, the company in Bangalore, has a very strong library using Indian blood samples from different parts of India. So there are definitely libraries with different CDRs. Yeah, back to you, Ashish.
Thank you. The next question is for Dr. Jain. How can we build thriving biopharma innovation R&D pipelines in a new dynamic environment?
Am I audible?
Yes.
Yes. So I think thank you for this question. It's a much bigger question. Maybe Naren can also add into that, but I'll begin. So biopharma innovations are largely limited in the country. The primary reason is, again, it's a Me Too market, which we are focusing on. When you are looking into biopharma understanding, there has to be government which plays a larger role, where government can build a robust entrepreneurship startup ecosystem, which government is trying its best. The reason is we are not asking, I would say, relevant questions which are needed. So some of the major issues and challenges which we have, like development of clones, where we don't have not too many people around. Largely, we are dependent on thermos or So though I'm talking in Denher seminar, but the group like Denher, which is largely or group like Thermo or Merck, we should have more Indian counterpart to these companies, and this is largely needed. Development of resins, development of clones, development of consumables, and then that will allow a system which will actually we will be able to do drug discovery. So as a CRO, I think a lot of drug discovery has also been outsourced to India. But as an inherent, because the cost of development is extremely huge, the chance of failure is and the risk is very high. These are some of the reasons. Innovation is something which require significant amount of support and also significant amount of um, entrepreneurs who have now established industry. The onus is on both the side. The young crowd, which is coming out from academia, and can take these risks and develop certain molecules. On the other side, there has to be significant amount of support from government and society, which can actually allow these innovations to happen. But the need is high. Yeah, that's all from my side.
Okay, thank you. The next question for Dr. Niti. This workflow is about the MAB, and you have nicely integrated the development to formulation steps. Do you have a similar solution on a bigger scale, and where you have a connected manufacturing process? And also, apart from MAB workflow, are you guys focusing on other technologies?
Thanks, Ashish, for the question. And thank you for the monoclonal antibody workflow appreciation. Yes, absolutely, because we work along with the customers and the commercial need. So that's why we made this monoclonal antibody workflow. Apart from this, we do have a vaccine workflow, which is on the similar lines, which is for the protein vaccines, because as of now in India, we have the protein vaccines. And also we have the gene and the cell therapy workflow also. And as I say, at our Danher, we always believe in continuous improvement. So we would be making more workflows to align with the needs of our customers and to have more innovative technologies in future. And answering the second question is, do you have a connected manufacturing process? So yeah, we do have the idea to injection, which it is called. So right since the inception of your cell bank to vial filling, we have that whole manufacturing process being covered. So we do have that. Back to you,
Okay.
Ashish.
Yeah, thank you. Thank you very much. We are running out of time. So we will end the session today. And I would thank all our participants for joining us. And I would like to take this opportunity to thank our esteemed speakers for taking out time from their busy schedule to be a part of this exclusive webinar, which surely has been engaging and enriching for all our participants. With that, we conclude this webinar on pharma possibilities of therapeutic monoclonal antibodies in partnership with Danaher Life Sciences, brought to you by Bluetech Media. Thank you, everyone. Thank you for joining us.
Thank you.
Thank you, Ashish. Thank you, Dr. Narayana. Thank you, Dr. Narayana. Thank you, everyone. Thank you.
Thank you, thank you, thank you, everyone.
Yeah.